Hey, Amy here, back with part 2 of section 11.2, in which we are looking at Mendel's crosses. So this is part 2. We want to continue talking about some of the crosses Mendel did and some of the more complicated things and how we also compare that to our modern genetics. Okay, the dominant and recessive alleles represented by DNA. Okay, so those alleles are represented by DNA. So our sequences, like remember the A's, T's, G's, and C's, that code for proteins. In other words, Mendel called them factors, but we now know today those are DNA genes, sections of DNA that code for proteins. Okay, the dominant allele codes for a protein that we often will consider the normal gene function. And the recessive allele often represents a loss of function. Okay, so a tall versus short. Tall causes the plant to grow more, whereas short, there's that loss of function. It doesn't grow as high. During meiosis 1, the homologous chromosomes separate. We looked at that a little bit in the last video about how when you've got maternal chromosomes and paternal chromosomes, how you might have big T, little t, and how those are all separated during the formation of gametes. Meiosis 1 pulls the homologous chromosomes apart. Meiosis 2 pulls the sister chromatids apart, so that in the through the process of meiosis, Mendel's law of segregations, that's why there's only one allele for each trait in the gamete. Okay, each gamete would be haploid. So you'd have big T, little t, and then you get an equal chance of them combining with the gametes from the other parent. One thing that Mendel did that we can do as well is do a dihybrid. Okay, di meaning two. So true breeding plants for two different traits. For example, tall and, plant height, tall, short, and P color, yellow or green. Okay, so you start with true breeding plants with two different traits. The F1 showed dominant for both of those traits, and then he allowed the F1 plants to self-pollinate, and he found this whole like a 9-3 to 3-1, if you remember from Bio 1, where 9/16 were dominant for both traits, 3/16 were dominant for one trait, not the other. 3/16 were dominant for the other trait, but not the first one, and 1/16 were recessive for both traits. So he formulated the law of independent assortment. This just said when you're following two or more traits, those traits segregate independently with the factors from the other traits. So you can get all possible combinations of gametes. And this is where I mentioned in the last video that Mendel was lucky and that none of the traits that he picked were linked. None of them were on the same chromosome. So did he luck out? There's some controversy. Some people say he did, but then he didn't study those traits. He threw them out. I tend to think that, hey, he was pretty lucky and he got 7 traits on different chromosomes. And so he figured out independent assortment, that those traits will assort because they're on different chromosomes. They will assort independently of each other, and you get an equal chance of all those different combinations of traits from the two parents, even if you're following multiple traits at once. And again, P is the parental generation. Just a reminder before we look at these crosses, F1 is the first generation or first filial, and F2 is the second generation or the second filial, after you let the F1 parents self-pollinate. Okay, this is a dihybrid cross that was done by Mendel. And he takes the P generation. You take pure breeding for two traits. This one, in this case, we're doing dominant tall plant and big G, green pods. And over here, this would be tall plants with green pea pods. Whereas this plant over here is recessive for both traits, short with yellow pods. Okay, and what happens in the parental gametes? You see that the you get an equal chance of getting either one. Well, in this case, they're homozygous for both traits. So what is it going to pass on? Well, this parent's going to pass, we don't have to write them all out, because it's going to pass on a big T and it's going to pass on a big G. That's the only thing it can pass on. This one, we could write them all out, but it's going to pass on a little t and a little g. So when you combine them, you put the t's and the g's back together. All of them are going to be, it's kind of small there, big T, little g. So what did he find in the F1 generation? 100% showed the dominant trait for both traits. So they were 100% tall with green pods. Okay. Now, when he let them self-pollinate, okay, let's look at the possible gamete combinations, okay? So if we cross big T, little t with big G, little g, and we cross that with another plant, big T, big G, little g, look at all the gamete possibilities. This big T could be passed on with this big G. Okay, so we see that right here. The big T could get passed on with the little g, big T, little g. The little t could be passed on with the big G, or you could get the two recessive alleles. Okay, so there's actually 4 possible combinations of alleles in the gamete. And so along the top of our Punnett square here, we have those 4. You're crossing it with the same thing. So here's our big T, big G, big T, little g, big G, and little t, little g. Okay? And so this is why we get that 9 to 3 to 3 to 1, 9 to 3 to 3 to 1 combination. Okay? Because if you add them all up, a 4 by 4 square, there's 16 total possibilities. Okay, so if you look here, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, if the yellow background, 9 of them are tall and green. Okay, and then 1, 2, 3, 1, 2, 3 are tall with yellow. And then 1, 2, 3 are short with green. And then you've got 1/16th chance that your plant would be recessive for both traits. Okay, so there is genotypic ratio, but we don't worry about it because it would be very complicated. Okay, for a dihybrid cross, we just often talk about the phenotypic ratio of being 9 to 3 to 3 to 1. Ooh, that's a rough arrow there. Okay. This slide is just relating to how this would look in meiosis of forming these gametes when you've got 2 traits, okay, A and B, okay, and how that would line up. Okay, now independent assortment says, okay, after this goes through replication, you'll actually have big A, okay, and then, sorry, here's the A's, big A, and then a little a, big B, little b. Okay, so you're actually going to have 4 pairs of chromosomes, 2 pairs of chromosomes, 2 of them are homologous, 2 pairs of homologous chromosomes. There, I said it right. Okay, now how can this line up? Well, notice during meiosis 1, during metaphase 1, that you can have, here you have maternal on the left, big A, little a, okay, paternal on the right, paternal big B on the left, maternal little b on the left, or on the right. And so when you divide them out, you can have big A, big B, big A, big B, little a, little b, little a, little b. Okay, but notice some of the time that might actually switch. So you've got 4 things. They can go this way, or 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 this way, this way, this way, this way, this way, this way. Okay, so you can see during meiosis 1, Here they are lined up in a different assortment. And so this assortment down here is going to give you the big A, little b, or the little a, big B. So you get all the possibilities, big A, big B, little a, little b, big A, little b, and little a, big B. There's your 4 different possibilities. Okay, so you get all possible combinations. This is independent assortment and segregation. They're being pulled independently of each other, the 2 traits, because they're on different chromosomes, and how they line up in meiosis 1, okay, at metaphase 1, you get all the different combinations. And when you follow that through meiosis 2, you get all the possible gamete allele possibilities over here. Oftentimes for Mendel's crosses, and we have been already, and you did in Bio 1, you're familiar with Punnett squares, named after R.C. Punnett, was kind of named after him, who kind of used these things. It's just, again, drawing that box, and if you're crossing, you're crossing 2 heterozygous tall plants, we can just draw a box, and we can divide it into 4. Oftentimes you'll see a little line up here where you'll put like female on one side, male on the other side, and you put the possible gametes on the outside. Okay, so possible sperm on the top, egg down the side, and then the possible in here would be the possible zygotes. Okay, so you'd see all possible zygotes, and we just fill it in. Big T, big T, big T, little t, big T, little t, little t, little t. And then you can count them up, and you get your phenotypic and your genotypic ratios. The Punnett square, again, it just kind of organizes it for us. It makes it easy to calculate probabilities of the genotype-phenotype ratios. On the next slide, we'll show, the Punnett square, the chance of getting A, 1 out of 2, little a, 1 out of 2. Okay, so the offspring, we use the sum rule and the product rule. Okay, so your chance of getting big A is 1/2 times 1/2 gives you, that's the product rule. It gives you 1/4. Okay, chance of getting that 1/4, 1/4, 1/4, and we'll see that here on the next slide. Okay, we're just showing 2 parents. Okay, that have big A, little a, big A, little a. Okay, and normal pigmentation and lack of pigmentation. So you have 2 parents with normal pigmentation, little a would be what we consider albino. You put here, they have the male, the sperm down the side, so dad can pass on a big A or a little a. Mom can pass on a big A or a little a. And you just notice that there is 3/4 a chance of being normal pigmentation, 1/4 chance being albino. Very easy to calculate because the Punnett square kind of lays it out there for us, and we can just count up how many out of how many total. 3 out of 4, 3/4. And so that gives us sort of a ratio that if you'd look at their family of 5, you could say about, or a family of 4, about 3/4 of the kids are probably normal, but they do have a 1/4, so probably 1 out of every 4 kids they would have would be albino. Is that what we always see? Nope, this would just be predicted results. And when we do our genetics lab in class, you'll see that our actual results don't always match perfectly our predicted results because of the laws of probability and the laws of chance. Last thing is just talking about test crosses. Individuals with the recessive phenotype always have the homozygous recessive phenotype. One thing I use in Bio 1 a lot as an example is sheep. Let's say I have a sheep. The daddy sheep is a ram, and we know that black is recessive. So if I have a black ram, I know that it's going to be little b. Okay, actually we shouldn't use b's. Okay, let's say black is dominant. So let's say I have a white ram. I know that it's little b. Okay, because that's recessive. If it's recessive, it has to be homozygous recessive. But let's say I have a dominant. Let's say I have a black ram sheep. Okay. Now we don't know we know that one of the alleles has to be dominant because it's showing the black wool. But do we know what the second allele is? Is it big B, big B? Okay. Or is it big B, little b? Okay. We don't know which one. So how can we figure that out? Okay. We will do a test cross. We cross it. We will cross this with a homozygous recessive. Okay. We cross it with homozygous recessive, and then we can look at the results and see if we can figure this out. And I'll show you this on the next slide. Okay, so this one, again, instead of using sheep, this is just saying, let's say I have a tall plant, and I don't know if it's big T, little t, or big T, big T. You cross it with short plants, and you look at the results. Okay, if, for example, you get all of your offspring are tall, let's say 100% of your offspring are tall, and you count maybe hundreds of plants, or maybe you count 20, 30, 40 plants, and in your cross, big T question mark times little t, and they're 100% tall, most likely your plant's going to be homozygous or a purebred. Homozygous tall, because it's only passing on a big T, so all of the offspring are going to be tall. Let's say you cross it with 20, 30, 40 plants, and you get about 50% tall, but you get a recessive phenotype show up. Okay, this tells you to get a recessive phenotype, you have to have two recessive alleles. Okay, so that means that my tall pea plant has to be heterozygous or a hybrid, because it has to be able to pass on that little t in order to get a short plant. Okay, so back in the case of my ram, okay, if I've got a black wooled ram, I'd cross it with a bunch of white wooled females, and I'd look at my baby lambs. Okay, a couple months later, when the ewes start having hopefully triplets or quadruplets, so that Hammy can make a lot of money selling them at the auction. We look at those lambs, and if the lambs start coming out and the lambs are all black, okay, if I have 100% black wooled lambs, then I know that I probably have big B, I probably have a purebred ram. Okay, if it's lambing season, and I did this cross, I crossed them all with white-wooled females, that first lamb that comes out that would have white wool, I would know right away that my ram is actually a hybrid. Because to get a white-wooled lamb, the ram would have had to have a little b or recessive allele to pass. And as lambing season goes on, I'd probably notice about half my lambs would be white wool, and about half my lambs would have black wool. Then I'd know, okay, this is a test cross. Without doing expensive genetic testing, you're just crossing your unknown dominant phenotype with a recessive phenotype, and looking at the offspring, you can figure that out. And you can actually do a two-trait test cross. Let's say my plants are tall, and they're tall, and thinking back to the example we worked earlier, they're tall and they have green pods. Well, are they pure tall, pure green pods? How do I know that? Again, cross it with an individual that's recessive for both phenotypes, for both traits. So I would cross my tall green plant with a short plant with yellow pods. And if you find about 1/4 of each, 1/4 tall green, 1/4 short yellow, 1/4 tall yellow, 1/4 short green, I have 1/4 of all the combinations, okay, then I know it's heterozygous for both traits. If I happen to get a 1-to-1 ratio, let's say I get 1/2 is tall and green, and one half is tall and short. So they're all tall. Then I know my height gene is homozygous dominant, but then my pod color is heterozygous. So if you get a 1-to-1 ratio, then only the one trait is heterozygous, the other is homozygous dominant. If you get a 1-to-1-to-1-to-1, then you know both traits are heterozygous. And so by doing these test crosses, again, we can figure out by what we're crossing and our results, the offspring, we can know the genetic makeup of our